name's Elise. I'm from How Do We Meetup AKL. Who's been to one of these before? Oh, oh, so many people, which is awesome. It's so good. Obviously, you had a great experience or liked the first time you came and you've come back, so that's awesome. So yeah, if you haven't been before, what happens is we'll have 3 speakers. We'll chat kind of one after the other. There'll be a short break in between. And then at the end, anyone who needs something can come up and pitch, and you have 1 minute. So people have got jobs, they've found employees, they've got all kinds of cool opportunities. I even went and gave a lecture at the university because of someone getting up and talking who's here. Who university lecturer is here? Yes, there. Yeah, so you had some great success pitching. Yeah, so if there's anything you need, you're definitely in the right place to come up and pitch for a minute. So that'll be at the end, and I'll definitely give you some warning. So we couldn't do this without sponsors, obviously. We've got Hudson Gavin Martin, Aunt Shirley here. She's a partner at law firm, amazing. They are super experienced. So if anyone has any legal questions, Hudson Gavin Martin actually give free legal advice to How Do We Meet Up people. People have utilized it before with success, so that's awesome. We have Blender here. It's the Blender guys. Blender is like product development and design. So they're there for you to chat to if you have any like product development questions or anything like that. And Blackbird Ventures is a venture capital firm. I don't think anyone's here from them, but yeah, feel free. They're super open if you want to hit them up as well about anything. That's perfectly fine. And then we're also sponsored by the event space Grid AKL. So yeah, super lucky. Cool. We're gonna crack in. David is going to get the slides up, and we're talking to Ollie here from Mint.
Hi everyone.
So
do you want me to use that mic?
Yeah. Ooh.
Hello everyone. Hopefully, can everyone hear?
Yeah.
Close. Good.
What is Mint Innovation?
Great question.
And who are you?
So my name's Dr. Ollie Crush. I'm the Chief Scientific Officer at Mint Innovation. And we are a small company out in East Tamaki that you can see behind me here on the slide. And we've built a process for taking weird and wonderful feedstocks that contain some metallic value that's usually not being utilized to its full potential, and using that as a feedstock for a process where we recover metals from them, like copper, gold, that sort of thing. So
when you say feedstock though, you don't mean like animal feed?
I haven't tried that yet, but no, I mean things like mining tailings or automotive catalysts or catalytic converters or old circuit boards, electronic waste. So feedstock, sort of the very generic term for it, but the process we use to recover it is very agricultural, so it does kind of tie into the animal thing.
Cool. So what? Yeah. How does it work?
Well, I've got a few slides.
Oh yeah, let's
walk through if you want.
Go through.
So I guess tonight's a bit of a biotech theme. So I've tried to focus a little bit on the biotech side. That's my background. I did my PhD in synthetic biology. But turns out with a process like this, there's a lot of chemistry involved as well, a lot of process engineering. We've got a couple of process engineers at the back there, Edwin and Marlo. They can answer all your questions.
Could you put your hand up?
I'm just the figurehead
here.
Oh, awesome. That's what they came for. They really like talking to people, so don't be shy. Anyway, so I'll focus a little bit on the biotech side of things and how we use microbes in our process to recover precious metals, but also just wanted to give people a flavour of our journey to date as well, because this is our 5th anniversary this year, and I, for one, am surprised we've made it so far. So hopefully I can share a few stories with you.
Cool.
So I did have a clicker here, didn't I? Let's just go through this. I'm just going to skip through a bunch of the stuff here, apart from this bit here. So why our first feedstock we focus on at Mint is electronic waste. Why is that? It's basically because you've got all these electronics going to landfill each year around the world, something like in the order of 50 billion tons worldwide, and they contain circuit boards, and circuit boards have valuable bits and pieces in them like copper and gold, platinum group metals, things like that. So if I just rip through a little bit here, how does this relate to biotechnology at all? Essentially, what we've come up with is a process for taking the bit that doesn't really matter from the board here, the plastics and ceramics, and focusing on the tiny little bit that really has the value here and here, and using microbes to help separate the dross from the valuable part. So we do this through a process called biometallurgy. So this is basically chemistry and biology intersecting using microbes. And it's commonly used around the world already for things like the oxidation of gold mining ores to basically break open the ore, expose gold to make it more amenable to traditional leaching technologies like cyanide, which by the way we don't use, or through really low-cost heap leaching of copper from low-grade ores. This is a picture up there from Chile, a basically massive mound of copper sulfide containing ore, where they put these microbes on there like this acidothiobacillus spheroxidans that's able to basically dissolve copper out into a little leachate, and they have these big piles on plastic sheets that collects them, and then they can electrowin the copper out of that solution, and off you go. So microbes are doing a very low cost job here in mining. But for us, what we're doing actually is we started off looking at using the microbes to do the dissolving of the metals from the circuit boards themselves. It didn't quite work out. So we focused more on using the microbes to help concentrate and purify things like gold from a very dilute and heterogeneous solution. So lots of other metals floating around in there, very hard to separate. So essentially, in a nutshell, we dissolve the feedstock, put our microbes in it, go in there and act like a functional sponge to pull out the metals that we want. And then because they've got a mass, we can recover them from the solution, and we purified and concentrated the metallic value up in one step. I'll skip through this stuff here. And then maybe I'll shut up for a little bit and you ask the next question.
I feel completely out of my depth with this topic.
That's okay. I've had 5 years.
So yeah, cool. I'm like, oh my God, like, how do I pick a question out of that? What does the journey look like? How did you start?
Yeah, so basically we've been going for just over 5 years now. And the journey was a little bit of a sort of, I guess, second time round story. So that's a picture of my co-founder and CEO, Will Barker, and I, when I had less grey hair, and Will had just come out of another biotech story called LanzaTech, which were taking carbon monoxide gas out of steel mills, out of their flue gas emissions, and feeding it to microbes. The microbes used as an energy source and made ethanol or other more valuable products. And that's still going strong, but it moved offshore in 2014 to Chicago. And they've grown to a team of maybe, I don't know, 120 in Parnell, and some of their staff stayed behind. Will was one of them. And so he'd been on this sort of this growth story with the biotech company before and wanted to do it again. I just returned to the country from doing my studies overseas, and sort of met up, networked, and then we just sort of worked on this. He pitched me this crazy idea, and then here we are. So started off basically big borrow steel in a fume hood in this old lab in Parnell, and this is our employee number one, Reese, on his first day doing some bucket chemistry, literally, and that soon outgrew the space that we had. So the lovely thing about this place in Parnell was there was lots of empty corridors and basements. So we decided to get the jackhammer out and build a lab downstairs, bit of a pilot facility to prove our process. Raised a bit of money to do that. That all worked out. Then we could scale up further to basically lease a warehouse in East Tamaki. So this was a couple of years ago now. Nice empty pad. And then built a bigger version of what we've done in the basement in Parnell there. So by this stage here, we've gone from taking sort of 20, 50 grams of ground e-waste to taking a ton at a time and putting it in tanks that were sort of 6,000 litres as opposed to
1 litre beaker. So yeah, that really does simplify things. There's a lot of grunt work that's gone in between now and then, but that really is the journey to date, apart from the fact that this year we are trying to build our first commercial facility over in Sydney. And so now we've got an even bigger warehouse to fill. The downside is it's in a red zone in Western Sydney at the moment, so it's a bit difficult to progress anything right now, but hopefully that changes soon.
Where do you get the waste?
So we are certainly not trying to enter the waste collection business. There's lots of people that do it already. So we're basically seeing ourselves as a part of the recycling chain. So there are people that already collect old computers. They've got far more value when they're refurbished and sold and reused as functional devices as opposed to metallic value. But eventually they break, and there are people that will be stripping parts out. The casing, steel, the copper wiring, that's all easily recyclable. But all these circuit boards, they just end up putting in a pile, and sometimes they get sent to the right end of life place, like a specialized smelter in a couple of parts of the world. Other times they end up going to developing nations where they're essentially put on a bonfire and metals recovered that way to the detriment of the workers' health and the environment. So actually the very short answer is we partner with people that are collecting this already.
Right. Okay. And is it from New Zealand? At the moment, it's been from New Zealand, hasn't
Correct.
it?
Yeah. So one of our collaborators is an outfit called Remarket IT down in Wellington. They have a lot of corporate and government contracts for recycling and disposing of IT equipment. So they've generously partnered with us to send us material to work with.
Who keeps the precious metals?
Well, at the moment, we get them, sell them, and return the money to Remarket. This facility that we've built behind me here, I've got a few more photos down the track, but they it's a demonstration facility. So essentially, we're proving the concept works at a scale that is using the same equipment, the same sort of volumes that you do commercially, but rather than rushing headlong and building, an ideal plant from very limited data at a pilot scale, we've basically built this giant prototype. We've modified it God knows how many times, and once we've got that settled, which we have now, turn that into commercial design and actually pour the money into it.
So when it's commercially running, how do you think your business model is going to work?
Yeah, so it's an interesting one. There's a couple of options. We can either buy the waste outright from the recyclers, and then they get cash payout and they go away happy. Or we can work as basically a service provider, toll refining. So essentially we hold the material for them, recover the value, take our cut, and then return the metal to them. So I think what we're going to do in the first instance is not try and rock the boat. We'll just do exactly what the industry is used to, so our customers don't have to think any different until we've basically got a foothold in the market.
Cool. And so the journey, what your roadmap moving forward, what does that look like?
Yeah, so I'll just skip through a bunch more stuff showing this works. It may have stopped. Maybe just the next one. I've got a few photos end to end of the process. I don't know if people are interested in seeing a bit of a picture story.
Yeah.
Okay.
Definitely interested. Has anyone got a joke in the meantime? Jason, you got a joke? Or the button.
We've already done that to a couple at work. The thing about a startup is that I'm still used to existing on the smell of an oily rag. So the first projector we got that was brand new was from Kogan. I don't know if anyone's bought anything from there. $400, great price, really shitty outcome. But hopefully I can show you what we did with it. So how this whole process starts is the BioTech part is really here. And that's where we started off in the labs, proved that really quickly. And then it became, how the hell do you actually get from where your recyclers are to the point that we want it, and then out the door. And so that's been a lot of effort. So we're more chemistry and engineer heavy. So the first part is we receive these boards here. They've got a whole bunch of copper in them, and the layers between the ceramics as the conductive tracks, and gold in the bond wires and some of the integrated circuits. I'll skip through this text, but this is our milling machine here in one of its frequent breakdowns. It's a hammer mill, so essentially imagine 16 hammers flailing around at 10,000 RPM and throwing something into it like a shitty Kogan projector, and you get the idea. What comes out the other end is basically a sand. So we do that because we need to make sure that we've got a great surface area of all the metals to be able to do the chemical reactions with. So it's a two-step leach. The first approach, and again, this is some photos from some of our scaling to date. So this is our 100-litre pilot reactor. We basically leach out things like copper, iron, nickel to start with, and I don't know what that photo was. And you basically mix it all up in a reactor there, and you separate it. This is a glorified cheesecloth, just to filter out the solid sands and the dissolved liquids. And again, that had some frequent issues because it was built by people that weren't engineers. And then we get to a demonstration plant here where we're scaling up the same process. So now we're sort of feeding material into a big 6,000 litre tank here with a screw conveyor, a big proper cheesecloth assembly to separate solids from liquids. And at the point where we've got the copper dissolved in solution, we can basically put in an electrowinning cell and pull out these lovely plates of copper that essentially is what they call 4 nines, 99.99% pure, and you can sell that to whoever. So the lovely thing about this business model is once you get to a commodity metal, it's very easy for people to see where the value is. And then we get into the nice part, the precious metal leach. So again, it's just another reaction, bit of different chemistry going on here. But this is the bit here where, although we've done a prior leach that's removed a lot of metal, there's still a whole lot of metal left over. That's not the precious metal. There's still copper in there. There's still nickel, tin, just at much lower levels, but still at much higher amounts than the gold. So I should have said that if you're looking at your typical mining ore, you've got maybe 10 grams to 20 grams of gold per ton of ore you get out of the ground, whereas these circuit boards, maybe in the order of 100 to 200 grams per ton that you're processing. So a lot more concentrated. So what we're trying to do is get that dissolved and then feed. This is just another really crappy picture of our reactor, but inside you've got these paddles stirring around the ground powder and the various chemicals. And then that's when we add in these microbes. So this is a picture of one of the microbes that we've used. It's Cuprovitis metallogerans. And I should say that deliberately at the start of this, we looked at using GMOs, engineering bacteria to do this, which is my bread and butter, what I tried to do, but realized that as soon as we got out of the laboratory environment, this is something that Landsec had encountered, that going past that point with the regulatory environment for genetically modified organisms outside the containment of a small lab just gets very difficult. So we deliberately decided to target naturally occurring organisms, and as Jeff Goldblum said, life finds a way. And in many different parts of the world, there's lots of metal contamination, mines, things like that, even just scrap metal left around. Microbes usually don't like metals. It can be toxic, but it's a niche, and so some of them involve ways to detoxify the local environment. And so we've been piggybacking off what nature's been doing for a few many years. And so I just want to show this picture here because what this particular bug does is it grabs gold that, and before this project I never knew, but apparently gold does dissolve naturally in the environment and soil and is toxic to microbes around it. And what these guys do is that they will pick it up and basically reduce it into little nanoparticles. They have a capacity for doing that. Along the way, they usually end up altruistically dying to save their compatriots. And we've taken that to the next level, which you'll see in a minute. So these are recovered microbes here, loaded with gold, maybe about 1 to 2% of their mass is gold now. And that's brewing up the microbe. recovering them from our process at a demonstration scale. They're a nice purple vomit.
What does brewing mean?
What's that? Brewing?
Brewing.
Oh, so basically fermenting up the microbes. So this is a nice little reactor there to harvest the microbes. We do that separate from our process because it's essentially an ingredient for the recovery of the precious metal. And then it's a one-way trip for them because we put them in a kiln and basically ash them. And so at the point they go into there, it's just hydrocarbons and gold. And then you take them out and the hydrocarbons disappear. And you're left with it looks like that every time. A nice. They're very well trained. No, that's a 150 gram button of gold that basically represents what comes out of a ton of e-waste. So that's about, I think today's price is something like $12,000 or $13,000. And the copper on top of that would be another maybe $6,000 or $7,000. So you can start seeing there's actually some good value in these boards if you know what to do with them. So that leads me very quickly to probably your next question, which is the future.
Yeah.
And this is not pre-planned at all. So the idea for us now, I mentioned we're trying to build this plant in Sydney. So we've got our basically our plant, but the size of it really, it doesn't need to process a lot of material a year. So we're talking about 1,500, 2,000 tons of electronic circuit boards coming in. That's enough for a profitable plant. So it means that you can start having these distributed plants around the place that don't cost, hundreds of millions of dollars. So we've got a little facility in Auckland. We're building in Sydney at the moment, or we're building in Sydney at the moment. Raised a bit of money last year to enable that. Looking at the UK next year. And then it's all about where to next from there and what the business model involves. Is it franchising? Is it build, own, operate? Who knows? We'll get the first one out the gate before we settle on that. But in the slipstream of that, we're starting to look at other feedstocks in our process, and things like automotive catalytic converters with palladium, platinum, old industrial catalysts, other weird mining ores. And the thing is, now we've been around for 5 years. We've had a little bit of press. You start getting people from around the world reaching out to you and saying, "Hey, I've got this thing. Would you mind looking at it?" Or, I feel like my the person that I supply my material to is stiffing me. Can you have a look and see what's going on here? So yeah, we've got more work than we can handle at the moment. But yeah, it's nice. It's a nice high-level problem to have.
Nice. Do you need any help from us? Anything we can help with?
I could really do with like a clone of me. So no, we're all good. We're hiring a project coordinator in Australia at the moment. So if anyone wants to relocate there, it's a one-way trip, I'm afraid, with current restrictions. That's me. There won't be any danger pay, but no, I think we're all good. It's more, I think, from my point of view, I came into this with a little bit of dabbling in startups before, but it's the first one that's really stuck. And there's a lot of war stories and things that go along the way. So more than happy to chat with anyone that's thinking they're crazy enough to do this journey or to start on it, or they're already in the middle of it and can't get out. More than happy to chew on that, and yeah, just wanted to share the story. And every so often we do have open days out at Mint at the plant, so people can come along and poke and prod and talk to people. But as I said, the two gurus at the back there are the ones you want to talk to, Edwin and Marlo. They'll answer all your questions. So
cool. Wildcard question.
Go for it.
Just off the top of your head, in 5 years, would look like, looking back, would you have done, like, what would you have done differently in 5 years?
From now, or if I started again?
Yeah, if you started again.
Oh God. Geez. I think there's always this tension where you get comfortable and you've got a pace that you're going at, but you always need someone to really jolt you to move a bit faster, to really focus on what matters. I mean, my background is an academic, really like diving into things and understanding them and going down rabbit holes. And when you're doing this on a limited budget with limited time frames, and you've really got milestones you've got to hit, you really got to pull yourself back from that. So yeah, maybe there's a few things we could have done more efficiently, but otherwise, I think from the pace of company's development compared to somewhere in the States, probably a bit slower, but I think we're doing okay. We're still standing at least.
Cool. Awesome. Questions? Matt? So the question was, when's your next open day?
Well, we had one a couple of months ago, so I would presume it'll probably be next year at the rate we're going. But yeah, I'm sure if Elise wants to organise one for the hardware team, we can.
Cool. Yeah, let's do it. Okay. Yep.
I imagine there's still quite a lot of quite gross waste that comes out of the process.
So the question was, there's probably a lot of waste that comes out of the process. What do you do with it?
Yeah, great question. So, and again, that's part of the, when you look at the, okay, great, we've got the valuable bit, but then you've got all this other stuff there. You don't have a business unless you deal with that. Our process solutions, what we do with them is we've spent pretty much, as I say, a lot of chemistry involved over the last few years, working out ways to recycle them to really keep everything going around. The nirvana is having a process where it's essentially you've got an inventory of chemicals and you're just feeding electrons in and taking metals out. So that's what we're working towards. At the moment, we've got a few co-products we make, like gypsum that goes into concrete or drywall or whatever. The biggest bugbear is the rest of the circuit boards themselves, the plastic, ceramic, glass, whatever. It's a mix that take and get value out of. So we've got some collaborations underway to see about using it as a supplement for concrete manufacturing. It's got some properties that actually help cement, substitute for cement, so you can lower the amount of cement you have in concrete. But yeah, that's a bit painful. So the other option for that sort of waste stream is there's waste to energy plants in other parts of the world where you've got the plastic, you can get energy back, help break down other waste. But it's something we're still working towards.
Cool.
Do you think that this technology could really inform the manufacturers of the components to, add something in that's actually very cheap that actually helps the whole process at the end? So Apple goes and changes how they make stuff because they've bought your tech because they're going to help capture that value at the end of what they put in at the start.
Do you need to repeat that?
Do you think it caught that? They probably caught that. It's a great question, though.
Sorry for the microphone. Yeah, no, I think probably we're a little bit too far down the value chain for that. So I know Apple has invested a lot in programs to take back devices and have robotic picking of components off boards and things like that, which is probably more, again, trashing it and turning it back into the raw metal is probably, that's like the right down there in the value chain. So probably not going to inform how they design and sell circuitry. But certainly we've had interest from a lot of larger companies about what we can do to help with their responsibility for end of life recovery from boards. Really, this product line that we're focusing on now ceases to exist in some years' time because everything's reused and obsolescence is not as big a thing. But it is what it is for the time being. And as I said, you've got 50 billion tons of this stuff being discarded each year. So it's it's a problem that needs to be tackled from a lot of angles.
Yep.
How much e-waste do you need there? How much e-waste do you need for that 150 gram coin?
The question was, how much e-waste do you need?
About 1 ton.
1 ton.
Yeah, where do you get the microbes from? Do they keep growing or start going in?
Oh yeah, so in the process I showed you there, we basically have a seed stock. We brew them up from that and then harvest what we need and then put that in the process. And then it's a one-way trip for them. My original brief was to get microbes to poop gold, and I couldn't do it, so it's 30 minutes.
Yeah.
Have you tried selective brewing, or is that not feasible?
So the question was, have you tried any selective brewing?
Yes, we've done a little bit of that, and to be honest, with the way that they're brewed, it's not like a pharmaceutical grade process where it's really highly controlled. So they need to be basically the baddest guys in the tank. And if they're not, then that's what we end up harvesting anyway, whatever variant of it comes out. So there's a little bit of that inversion going on, but a lot of it's done mainly from, okay, where were these guys found? And I spent a few holidays going around New Zealand collecting samples from various old mining sites or refuges and things like that. So, yeah.
That's really cool. That sounds like Indiana Jones or something.
Yeah, but not so fun for the wife and kids. So.
Sav?
Have you come across any freaky microbes and why are they freaky?
Have you come across any freaky microbes and why they're freaky?
Not especially apart from the fact that they've got this weird property of detoxifying metals in their local environment. In a way, we kind of want to steer clear of the ones that are too fruity, because usually that means they have health implications if you're working with them. And so we want microbes that are basically what they call generally regarded as safe. So they're not going to, last thing we want is people getting sick using this process. So, yeah.
Cool. Hannah?
I'm very interested in the fact that you raised so much money. It sounds, maybe I'm wrong, but it sounds like you're still deciding on a business model.
Hannah said super interested in how much money you raised, and it looks like you're still working out a business model.
Yeah, so it's probably because I'm the wrong guy to be answering this question. So I'm the science officer with starry eyes. No, business models that basically they're ready to go. It's just we want to experiment with which one will stick, and time will tell. And the nice thing is that we can start with one. If it's not quite working, then we can switch to another one. But they all involve processing this waste and getting copper and gold and other metals out of it. So yeah.
I suppose you can just show them the gold nugget.
That's very, yeah, exactly. They get what we're trying to do very easily.
Yeah.
Could you dig up a landfill?
Question was, could you dig up a landfill?
Yeah, no, we've been approached about that, and I've thought about it before. I think the issue with that is that you're starting to get down in that concentration where you want a landfill that's got a particular type of material in it, and if it's all a bunch of too much other metals and things like that. I mean, we've got potentially about 30% metal content in the circuit boards, but if you're dealing with something that's 60, 70% metal content, then this process might get overwhelmed. So I know there are academic programs around the world that are looking at injecting microbial cultures into landfills to get them to basically dissolve and mine in situ. But it's very early days, and I think something like that, trying to control that culture of microbes in the wild, that's very ambitious. So yeah, but if you've got a secret landfill that's got something in it you want to, we can talk afterwards.
Okay, maybe a couple more questions. Yeah.
I'm curious as to whether there's scope for you to cooperate with gold miners, either in terms of enhancing their output or in terms of getting rid of your waste.
Question was, could you work with gold miners?
Yeah, definitely. And when we started out, we thought we'd be working with mining companies from the get-go, but initial sort of market analysis looked at their lead times for purchasing things, for making decisions, everything else. And when you're operating on a sort of initial budget of $20,000, then you probably don't have 5 years to sort of deal with them. So yeah, it's something that we're going to be looking into, I think, and there's a bit of opportunity there, but there are also a number of other people looking at the market as well. So it's yeah, we'll see.
Yeah.
Is there any carbon release from
What was the question?
Is there any carbon release from
Is there any carbon release from the process?
Yeah, so the bit where carbon's released is when we basically ashing the biomass. So as I said, it's hydrocarbons. We burn them to get rid of the hydrocarbon part and release and coagulate the metal that they're containing. But I should have said that you've got a ton of this material. By the time you get to the biomass being added, you're talking about a couple of kilos of biomass. So you're looking at sort of a 500x plus concentration of this metal onto this. So the amount of, you're talking about, you know, a home fireplace sort of level of emission at that point.
Okay, 2 more questions. Yep.
Are you processing electronic waste locally, or are you getting supply from overseas as well?
So the question was, are you just processing waste from local, or are you getting it from overseas as well?
Just locally at this stage. So our partners down in Wellington, they send it up to us. But there's a number of international conventions like the Basel Convention on the Transnational Shipment of Hazardous Waste, which is basically trying to stop people from sending this sort of material around the globe, especially because a lot of it ends up in places where it's dealt with inappropriately. We're really mindful of that, and hopefully this distributed plant that we're trying to build will enable you know, each country or each large city to have its own processing capability for its own waste that it makes.
Cool. One more question behind. Yeah.
Have you considered batteries? Can you recycle batteries?
Have you considered batteries? Can you recycle batteries?
Yeah, no, that's something we get asked a bit, and we haven't dedicated an R&D stream to it yet. Haven't really looked at cobalt or lithium or anything like that. And again, that's part of the, it's not that we don't want to, it's just part of the reality when you're trying to pull yourself up as a startup like this. You've got to focus on something very narrowly to get revenue out the door to start with. And we, you know, we're still pre-revenue because we've got to build this plant in Sydney as our first commercial one. And so that will give us the oxygen to start pursuing other feedstocks. And the nice thing is, you know, our team is small, we're 30 people, but we're developing this in-house expertise to deal with all these feedstocks and processing and the engineering and the marketing analysis and all the sorts of bits and pieces you don't really think about when you start out. We've kind of built that capacity up. So it means that we're actually in a really great place to start adding new product lines, but equally, if we start diversifying our attention too much right now, then we're just going to set ourselves up to fail on all fronts. So yeah, it's coming, but not yet sort of response, I'm afraid.
Cool. Would anyone actually be keen to go on a field trip to visit Mint? Yeah. Oh, awesome. Okay. Well, I'm really keen to organise that.
Yeah, no,
if you'll have
I'm
us.
happy to host Marlo. That was his idea at Edwin. They were telling me we should invite everyone to come back. So
Awesome.
Great.
Call that tomorrow.
They said tonight.
Tonight, yeah, we can. Some people can probably do tonight.
We'll organise something, no dramas.
I'm keen to organise it, so yeah, I'll organise it and get it out to the database. Are you going to stick around for a while?
Yeah,
tonight?
I'm going to hear the other speeches. And,
cool.
I haven't even been yet, so I assume there's a success. So,
there probably isn't, but there is a countdown over the road, and yeah, it's probably all gone. Though, if you go out, the door will probably.
I'm joking. Don't worry, it's fine. I'll send everyone a mail.
Yeah, so the door like auto locks, so we're kind of locked in. So if you go out, just like tell someone and get them to let you back in if you go outside. Awesome, Ollie. Thank you so much.
No problem. Pleasure. Thank you.
Cool.
And,
thank
yeah,
you.
Feel free to
So we're talking to Helico Bio now. Hi guys.
Hello. Hello.
Hello. Who are you and what are your roles?
So just a quick introduction. It's Three co-founder here. I'm Iliam's CEO. Currently, basically responsible for keeping lights on, commercial strategy and operations.
Hi everyone. My name's Nikolai. I'm currently lead scientist at Helico. Sort of developmental biology background, and you'll get to see a little bit of what we get up to if VR doesn't do something, you know, glitchy for us later, but hopefully I'll show you what we're doing.
Hi everyone, I'm Xiaoming. So yeah, I'm the lead software developer of Helico Bio, and yeah, we get to hopefully get to show you soon what computational stuff we've been doing in the dry lab.
Are you getting a call? Oh my god! It's probably someone locked outside because the doors are like auto locked.
My wife. Shall I answer?
Put her on the screen.
Put it on. All right, so good.
Awesome. What does Helico Bio do?
We have a kind of a next slide from that. But before that, I just wanted to do a little quick disclaimer that this is our new logo. We're very excited about its ancient symbol called the seed of life. You find it in ancient monasteries actually around the world. But if you go on our website and it's different logo, it's the same company. We just haven't changed it. And the reason we changed it, well, we're really young, 18-month startup. We just downloaded for $10, and our lawyers said they're going to kill us. So we developed our own logo. But yeah, so this is the first time we're actually using it. So just
Oh my God, it's a
discrepancy.
world first for the logo.
World first. Yeah. So what we do, we are in very high-level statement. We're looking at plants as a production source of useful compounds, and we're currently focusing on pharmaceutical compounds, but we kind of put them as a sort of therapeutic compounds that could include food and vitamins, any type of basically intakes in your body. And our goal is to replace steel factories and large sort of pharmaceutical production facilities with just farming, basically low cost agriculture.
Awesome. How does it work?
Yeah. So
Actually, why plants?
Plants. It's really interesting because currently, if you're producer or designer, drug discovery designer of pharmaceutical products, you either go into kind of chemical plant production, or if it's biosimilar products, it's kind of giant sort of brewery plants. Oli was showing something, but basically multiply it like 100 times. It's very expensive to replicate, very expensive to design. So plants naturally, as we kind of all know, synthesize compounds. Sugar, salts. They're also been known throughout the history as a sort of natural producer of medicinal qualities. So kind of in combination of two, it's very good platform and almost natural progression for humanity to look at the plants as a source of production. But also not very easy to design because complexity of plant cell is quite high. So it's almost comparable to human cells. So, but they're very easy to grow. Obviously, they don't require building facilities. You can do it in either literally in the farming or maybe in confined environments.
Or for some people, the roof space.
Yep, roof space of billionaire houses. Yep. We had that concept, but it didn't go well.
That was a marijuana reference.
Yep.
Okay, I'm not saying nothing to do with me or anything like that. Okay, how does it work? This is kind of the one slide that very schematic explains how our technology works. So we look at target molecule, a specific basically compound that needs to be produced. We run it through computational synthetic biology pipeline that actually we're going to have a little bit of a demo. This is just one of the tools that we're using. And we actually managed to put a patent application for that in the States, which is very exciting. Once we sort of reach certain level of comfort with the computational model, then we move into wet lab experiments, and that sort of loop between in silico computer simulation and the lab work is basically how our technology, kind of core technology, works, and also where we think we are kind of in a unique position because there's plenty of companies, large companies around the world that do either or both, but they have more room in one small department than people probably in this room today, and it's very difficult to create innovation. So we have a very kind of short loop between wet lab and basically skill set as well, plant biology and computational work.
So once we reach to certain level of experiments that actually satisfactory, then we move to the stable and production. So basically, we're looking at the plants that naturally produce the compound through a GMO modification. So again, Mint didn't want to use, microbes, GMO microbes. We have no choice. We have to design basically the brand new plants that never really existed in normal environment before. So once that plant is produced, then we can move to production. But at the moment, this is where our basically work currently stops. So we basically will look at licensing of these plants to either countries or pharmaceutical companies.
Cool. What's the journey look like so far? How did you start?
It's actually interesting. So we have a fourth co-founder who is not here today, and actually he decided to go complete his PhD. And we've met maybe 3-4 years ago. It was a very interesting idea, although he has a little bit, the idea sort of shifted. And then about 2.5 years ago, he actually called me in the middle of the night. And I thought, well, it must be something very important. And he said that, he managed to synthesize a very small molecule in a tobacco plant that could be potentially comparable to commercial yield. So, for me personally, it was kind of a moment where I was like, oh, wow, so this is actually it's not a PDF company, it's not theoretical science. Actually, it's moved to practical results. And then we collectively decided between me and Wayman that his process was actually kind of his own way of simulating machine learning algorithms, so computational side. And Wayman approached Nikolai, and I approached Xiaoming. And actually, that's an interesting story. We're basically looking for a third co-founder, and we accidentally booked them together. So they showed up at the same time, which was very embarrassing. But after they, we all kind of left that meeting with the feeling that actually it was sort of some kind of magic happened.
Well, so you had the meeting with both of them? Yeah,
with both of them at the same time. And there was just a little bit of a real magic moment. So we decided to just form company as the four of us. And then we raised capital in the beginning of 2020, right before COVID, just literally 2 weeks before that. Our first power station computer lost forever in the mail during COVID. We moved into level 2, which is Outset Ventures. Great place. So, Mint came from, or still is part of Outset Ventures, and we're probably the youngest kind of resident there, one of the youngest residents there. And that's so we will raise capital, and then Outset Ventures built a very nice facilities for us as a wet lab, and we also raised capital again beginning of middle of last year from myself, Venture, and some other funds, and then we're kind of in between two spaces. So computational team sits in Icehouse Ventures, and guys mostly spend their time in close to the lab, but it's 100 meters walk.
Awesome. What's this slide? What's the slide?
So basically, we built this computational toolbox. So we're going to demonstrate it a little bit later. So it's basically machine learning algorithms to simulate plant biology.
Awesome.
So that's selection screen. So they're going to show a little bit of that at the end.
Cool.
Well, and yeah, BioLab mentioned. So we still waiting for our EPA approval to do this type of experiments in New Zealand.
So why do you need EPA?
Well, it's New Zealand actually one of the toughest, country in the world to do that.
Is it because you're genetically modifying, creating new plants?
Yeah, but it's all in confined environments. It's not a problem, but it's still sort of challenging to get it in New Zealand. we're working in our first commercial deal, basically client in the States, which is super exciting. We can't mention who, but it's an amazing opportunity. And they were just shocked to find out that we actually need to get this type of approvals just to do experiments in approved containment labs. But that's just New Zealand, unfortunately. So basically, the end. If you kind of summarize what we do uniquely from other companies that produce pharmaceutical compounds, usually from plants, we produce them in plants. So it's basically the stable line is that seed based. So it's basically the seed that you can get from us will be capable of naturally bioaccumulate. So it's called stable line. So this is our actual growth chamber in level 2. So we're preparing everything's kind of ready for the experiments to launch.
Cool.
We decided to go for our proof of concept first compound as insulin, mostly because it's really a large global problem. We also done a lot of early stage validation of in insulin, and it's not necessarily price that people in in US and EU think it is very expensive, but for different reasons. We're mostly focusing on the third world and accessibility of insulin. So we build facilities in Africa and Southeast Asia, a few countries that we're actually talking on almost national level. They don't really mind to have first of all cheap insulin. Secondly, might not have sort of Rolls Royce of insulin that Western world is accompanied to, because insulin came from originally from extraction from a pig's pancreas, and it didn't work always, but it worked 90% of the time. So it's not good enough for civilizations we know as where we live, but it's actually good for other places where people are just dying, literally. So it's a kind of different way to look at it. We don't think we're going to go for anytime soon for some form of FDA approval. It might happen within novel forms of insulin that may not have a conventional production developed for it, but that's sort of different. This is more probably different conversation as of now. But insulin is a huge problem. So we're tackling human insulin as a first compound, and actually, guys, will show you where we are right now. And there's hundreds of different types of insulins. Majority of them under strict licenses for global pharmaceutical companies. But also those four global pharmaceutical companies have a sort of tendency to exclude countries because it's not profitable to sell it there. And that's the countries that we're actually targeting.
So the roadmap moving forward is EPA approval.
EPA approval.
And focusing on insulin first.
Yep.
Cool.
And also closing this first super exciting contract, which is mostly antibodies, novel antibodies.
Nice. What else we got?
Got a bit of a concept.
Oh, cool. And so this is kind of a concept that we developed by biofarming. Actually, it's we thought, we coined the new, but it's actually exists. So it's literally our idea was to fit it into shipping containers. So one would produce about $3.5 million worth of insulin per month in a sort of rough estimates, and then other one would have extraction purification facilities. So and then you basically drop those at crisis. So we did a study with the refugee camps and a few other ideas.
Cool.
Well, it's just really more of an immediate future for us. So we're going to look at basically patent application in the States with the same legal firm to basically secure insulin in plant production, and then move to commercialization with insulin, or possibly with even already different partners.
Cool.
The future, we always thought about this kind of idea of, and then also sort of looking at a few competitors that we have in the States and in Europe. One is in Israel. They all have very strange commercial partners, and mostly it's sort of military and space companies. And we sort of wondered for a while what's going on, and then realized that actually it's probably a perfect idea because it's sort of like grow as you go. So, in our case, it will just, you take the seeds with you and you grow your supply when you're, you can really take a lot of supplies with you, submarines or in case of emergencies or global crises.
Cool.
And then I'll pass it to Nikolai and Xiaoming. So we jumped on this platform called Nano in the States. We found it really exciting. Oh, I'm done.
Is it working? Oh yeah, it's all.
Maybe we can take some questions and while we.
Oh yeah.
Yeah,
Give us a
questions.
question, yeah.
Questions while we set up. And actually, do you need any help? Do you need anything at the moment, like from us?
If I start screaming, but you can see it, we're behind already. That's a good start.
We having difficulties finding people in New Zealand with the job description that we're putting in. A few people said it's probably 3 and 2 already left. So it's basically synthetic biology, machine learning, protein design. So it's all difficult to find. So if anyone's looking for a role with a computational synthetic biology, please contact us.
Awesome. So what are we going to see?
What are we going to see? So this is Nano platform. It's basically a VR platform where you can assess almost instantly binding mechanisms, kind of designed for instant kind of innovation. They're actually quite successful. Usually pharmaceutical companies and outside innovation labs kind of using it. So we're using this actually kind of make an instance assessment of some of our pipeline. And I think they're going to be showing insulin, but I'm not sure.
Is this?
Are you guys going to talk or?
See, the thing is, when you put the headset on, the audience disappears and it becomes a lot easier.
Sure. Do you want to hold it up? Yeah, Nikolai.
Oh, am I? Oh my God, I'm on. So currently I'm logging in. What we're going to show you today, hopefully, is, well, to make insulin, you need to start with a precursor of insulin, and that needs to get processed into its mature form, which is an enzymatic cleavage event. And so you need to have something that really well, electrostatically and from a lot of reasons, sort of these two molecules need to find each other in space. And then one molecule needs to cut the other molecule in half. And if that successfully occurs in the plant, then the plant successfully creates insulin.
Nikolai, can you go back in?
Back into where?
Oh yeah, perfect.
Zach, come on.
All right, just give me one more second. I might have to exit to the lobby. Are you in there, Xiaoming?
No, I'm
This is like.
We thought this
Technology.
No way.
So is this like synthetic biology Sims?
Yeah,
pretty much.
Or what's that other one where people have like entire families and stuff? No, it's like Other World or something. Second Life or something. I don't know. So that was a question about.
No, Nikolai, now you have to be in VR and answering scientific questions at the same time.
Plants
Tackle
versus
that.
animals.
Not really an issue, to be honest. It's the same genetic code, and plants have an amazing capacity to produce human-like compounds. It's the same exact, the same sequence, the same makeup that you need to make insulin in humans. It's just the plants are going through that process. And actually, plants are a much closer environment to humans than bacteria are, which is where all of your drugs are made. So the more interesting thing is, why isn't this happening more like sooner? And it's quite interesting, given that fact, you really have to engineer bacteria quite a lot so that they can produce human-like drugs. Whereas with plants, the idea is that you can do a lot less engineering and still have high-quality human-like drugs. And so that's quite a motivation actually to like sort of be in this field so that we can do that.
What Nikolai is actually saying is once people take this insulin, they'll turn into a plant. Oh yeah, cool.
Just developing on what Nikolai was just saying. Totally get what you're saying about plants versus bacteria, but what advantages do plants have over yeast or algae, which are also eukaryotes and have similar biology?
Okay, so that's yeast versus algae.
Yeah, well, yeast versus plants. I mean,
yeast versus plants.
Every country on the planet just about has got a beer industry, so there's a lot of technology and skill around yeast. So I'm asking, genuine question, what are the advantages of plants over yeast?
Well, the answer is sort of multifold and depends how much you know about glycosylation events. But plants have developed enough complexity to be able to stand up straight, have multiple compartments, have photosynthetic apparatus different from nutrient acquisition apparatus. There is a lot of inherent complexity if you just look at a plant and then think of how small a yeast is. It's a single cellular organism. So there's certain components, or what I would call like micro machines, that only exist in plants that don't exist in yeast, and they're more complicated and made of more proteins, and they just have never been sort of evolved to form in the yeast, and they're possible of creating certain variants or forms of drugs that are incapable and sort of left behind in the current drug industry. It's like, we can't do that, so we won't do that, and we'll go through a long route to find the way around. And then in plants, it's like we might not have to go on that long route. Now, the other thing about plants is they have a series of compartments, and those compartments are really interesting to us because, once your bacteria is in the bioreactor, it's hard to pull apart all that colony, pull apart that population, and say, I want to affect only those ones and not those ones. Whereas in a plant, I can feed the roots sort of nutrients and feed the leaves, my factories, sunlight. And the leaves are kind of, well, the plant's already turned itself into different elements of the factory. And so rather than taking the whole plant, I could take maybe just the aerial parts of the plant and not have to process the roots. And so you're able to compartmentalize the production of drugs. You're able to do new drugs that weren't otherwise done. You're able to do very interesting things with effectors and say, how does this plant respond to this effector? Which is kind of a case in bacteria, but it has to be in the middle of a bioprocessing plant. So you can't just affect it a little bit. It's got to be incredibly controlled. And so we can have plants in the field growing human-like drugs, and we might be able to give them some sort of a signal to do something quite important at a certain time point. And to me, as a plant developmental biologist, that's really fascinating, really cool, that you can use plant physiology and plant development in collaboration with drug development and drug production, which is kind of a bit of my secret sauce in the team is thinking in that way and thinking, how do you use not just the plant cell, because people will tell you you could do plant cell culture, but I really like the whole organism perspective. And hopefully that's a few, but if you want more, we can keep going on. Why plants?
Okay. Yeah.
I can answer it much shorter. If you can grow your beer on your balcony, the line will be out of business in one week.
Question. Yep.
So what forms of machine learning is your company leveraging and how?
What forms of machine learning is your company leveraging and how?
Good question. So we are utilizing actually much more than machine learning. Machine learning is one aspect of, I'm not sure if you guys have heard of the recent developments by DeepMind with the protein folding suite. So to give you basically an overview, right now there was a 50-year basically challenge in biology, where this is a kind of a holy grail of structural bioinformatics. If you can predict a 3D structure of the protein from just its primary amino acid sequence. Now, the tertiary structure aspect of proteins has been, people have been saying it's solved, and it's true to a large degree. So that's, if we don't have, if we're designing novel sequences in plants, and there's no lab data for that, tools like that is what we will utilize. And Google's throwing, I don't know, $1.8 billion at this problem. So much more resources than obviously we have. But we are utilizing techniques like that to fully do our synthetic biology without spending years and years of development waiting for guest design, then iterating of wet lab, seeing the plants grow, etc., etc. That's what traditionally you would have to do if you wanted to do that. That's like the most, I guess, 60-70% of the machine learning part for that. It's utilization. The other aspect is we use a lot of molecular physics to do our structural stability, and that's a very equally exciting part of this whole platform. Unfortunately, the VI demo didn't work, but yes, what I've seen is one of our designed sequences folded into a 3D structure. It's never been seen before in nature, and using stability calculations to see how they would interact. Yeah, sorry, that was a long one.
Cool, no worries. Yeah, so we might just have like 2 more questions because we're running a little bit behind. Yep.
I was wondering, you guys are genetically modifying plants. I mean, and you say you keep putting it in a controlled environment, but I mean, like what I said earlier, life finds a way. So, like, what procedures do you guys have in place if some genetic material like leaves and crosses with other plants? Like, what sort of environmental impacts could result from that?
So the question was, what environmental impacts could result from your crazy inventions getting out into the wild, and have you planned for it?
Well, I'm not sure if can answer that question, but just the 2 points. First of all, we're still pretty close to EPA approval, but we have MPI approval, which is basically where you do the experiments, and it's extremely robust. So it's all there. So it's like a high security, as high as you can get. Any lab in the world kind of follows the national protocols for that, so they have all this set up. But secondly, I just want to mention that it's actually not really well known fact, but the current insulin in the market is produced by genetically modified bacteria. So in fact it's the first ever approved by FDA a GMO product in the United States. So when people sort of say, "Oh, it's GMO," I mean, well, the insulin that saves your relative's life is also GMO. So it's a GMO conversation is actually not really necessarily relevant. I mean, we just talk openly about it. But I mean, what happens if it escapes? Well, yeah, it might lead to disaster, but at the same time, there's plenty of GMO that actually is already grown and even in non-confined environments.
Yeah. Last question.
Just out of curiosity, are you making new drugs or just something that's, say, structurally similar to insulin? Because you say if you want to use AI, that means you need to trust the coding and everything like that. But if you're just like making similar drugs or anything, that means I guess the coding will be really similar, or is that using the same genome sequencing?
So
wait, can you just summarize that quickly?
So the question was whether we are making new drugs based on existing similar sequences, or we're totally. Oh, sorry. Sorry, did I capture that? Basically.
Sorry.
So the question was, are we making completely new drugs, or are we using something similar to what's existing out there? Okay, so yes, the class of drugs that is the biologics, not the small molecules. And we generally go for things that are already FDA approved. That's the current plan. The folding comes in the sense that the stability of the drug that we're producing may not means that you may not want to produce a final drug in the end. You want to produce something that needs processing, pre-processing. And that's where the novel sequence folding comes in. If you produce something that needs pre-processing, that doesn't have to be the final thing that the people take. That just needs to be something that stably exists in a plant environment. But that sequence can be anything, and that sequence may not have existed before. So that's where the folding of the novel sequences would come in.
Yeah.
We're getting there.
I know we're probably run out of time, so I'll just explain it very quickly what I'm doing and how we use this. So biological molecules are really complicated, and knowing how they work is super, super difficult without extra information. It helps to get really close to the molecule. And so here I can see a methionine, a cysteine, a histidine. So this is the active site of the COVID protease that without this protease, COVID can't replicate itself. So if you were to do something to these residues above me right now that stopped them from functioning, we'd be able to hypothetically stop COVID from functioning. In our case, something like this molecule is able to cleave insulin. It's also an enzyme. It's also a protease. So a protease is a protein-cutting enzyme. And so in our situation, and apologies we can't show you what we intended to show you, but I'm trying to fit things into these active sites. And so we got to see what electrochemical reactions are possible. And how an enzyme works is it creates a chemical environment that favors a certain chemical reaction. So if you have a certain small molecule or other protein that finds its way into this active site, it gets processed and the reaction proceeds. So if this was an enzyme that was able to cut the precursor to insulin, proinsulin would get stuck in this pocket, and then an enzymatic reaction would occur where different chemical groups would react, and you have this cleavage. And honestly, we're doing this on 2D computer screens for the last year, and now we've been pulling it into 3 dimensions. And just the ability to kind of like spin it around and really get close to what you want enables us to do a lot more interesting science. And you'll notice that, a lot of this looks like a cartoon, but it holds a lot of detail. So we've got meter-plated sheets over here, and we've got barrels going on behind me. So that insinuates, potentially. I've got a little alien flying around me in space as well, but that's fine. So there's features on this molecule that are really helpful to diagnose the problems. And then, I can't do all the impressive things I wanted to do, but I could have two different things docked together. I could look at all the variable ways that one protein will dock to another protein. And seeing the results as numbers in Excel spreadsheet tells you one thing, but seeing how they dock together in 3 dimensions really tells us a lot more about the story and enables us to do good science development.
Awesome. Okay, that is all we have time for these guys. I'm super excited to welcome Carmen. Carmen's the CEO of Helios Therapeutics. Carmen, what does Helios Therapeutics do?
So thanks for the introduction. So at Helios Therapeutics, we are producing medicinal cannabis.
Awesome. How?
So at Helios, we're the largest medicinal cannabis company in New Zealand, and we're vertically integrated. So we go right from seeds through the growing process, through an extraction process, a purification process. We make those finished products, and we also have our own GMP analytical testing labs on site. We've just achieved the GMP license to manufacture medicines. Some of you may have seen that last week, which is a huge milestone for us at Healius. It's a huge milestone also for the industry, and most importantly for Kiwi patients who've been waiting for these Kiwi-made medicines to come to market here in New Zealand.
Yeah, so what does that actually mean? Like, has up to this point medicine only been able to be imported?
So a little bit about the medicinal cannabis scheme, because a lot of people get confused around cannabis and the referendum, the recreational referendum that we had last year, the medicinal cannabis scheme and what that means. So the medicinal cannabis scheme came into act on the 1st of April last year. What we've had in market currently has been imported products that have been able to continue being imported under what's called a grandfather clause. But this medicinal cannabis scheme sets quality standards for cannabis here in New Zealand. So going forward, all products, whether imported or made here in New Zealand, would need to meet the medicinal cannabis scheme quality standards.
Cool. What do you got? What's next?
So there's a little bit more to medicinal cannabis than just growing plants. So like we said, we go right through seed to sale, but actually as part of our process, there are a few other things that we're looking at. Healius exists to bring novel therapeutics into market. So a big part of that is combining both the manufacturing processes together with R&D and also together with clinical trials.
Awesome. What next? What's your vision?
So our vision, thank you for asking. So there's a lot of stigma around cannabis when we talk to people, and I can already see, looking across the room, there are some people who are excited and keen and want to learn more, and some people who maybe
wanted a refund, they didn't get one.
Yeah, sorry about that. I guess the key thing that we want to do is to help people to understand that we are seeing cannabis as a medicine. It has huge therapeutic opportunities. There's over 100 cannabinoids that have already been discovered inside the plant. And these cannabinoids work together with the endocannabinoid system in your body, which all mammals have. So humans have it, as well as all mammals. It's a new medicine, but it's actually an old medicine as well. It's got a long history. It's got all sorts of reasons that we have this stigma around it. And what we want to do is to be able to create medicines that really help people to have a better quality of life. Believe me, I understand some of the stigma. I come from the pharmaceutical world. My background is in mechanical and biomedical engineering. I spent most of my career working in pharmaceuticals, and when I was first approached to come and work at Healius, I was like, oh, I don't know about that. Cannabis, is that really a medicine? But I have researched it myself. I'm still learning all the time. And we're still learning as well all the time. And that's why we talk about unleashing the potential, because there is so much research potential here, both around the cannabinoids, but also around delivery mechanisms to be able to bring novel therapeutics to market that can really, really help people here in New Zealand, but also all around the world.
So what's the journey look like? Might just skip ahead a little bit.
So what I'll do is I'll give you a few slides on the journey, but just pull out some of the key points. So we were one of the first movers in New Zealand. The idea was first thought up in the back bedroom of one of our founders, which he proudly still brings up whenever we have calls, and he's sitting in that back bedroom.
He's like, this is the exact spot right here.
This is the spot that I came up with the idea. But we started our journey 2017, but became Healius Therapeutics in 2018 when we did the first capital raise. The first capital raise raised 15 million, and right from the beginning, we have been privately owned with 100% New Zealand investors who really truly believe in our journey.
Where did your investment come from?
So our investment comes from a number of families in New Zealand. A lot of them have a personal connection to the health and well-being of New Zealanders, and some of them also have a really personal connection where they've seen the benefits of medicinal cannabis.
Thanks.
So there's a few other milestones up there. I'll let you read through them. This is our site as well, and we're in East Tamaki, just around the corner from Mint Innovation.
Nice. We can do a trip to you too.
Oh yeah, we'll do a combo.
Yeah. How would be keen?
Is there?
Oh, cool. Yeah.
I was wondering how long it was going to be till someone asked for samples. So
Yeah. Yeah, it had to come up. I
It had
wasn't
to
sure if
come
it
up.
would come up this early, but thank you.
it was going to come. So our product is a prescription product, so I can't offer samples. Obviously, the only way to access our product would be to speak with your GP or with your doctor.
Yeah, so I have had this question before. How do people access this? Because there is still illegal in New Zealand, but a lot of people don't realize that it is actually legal for medicinal purposes.
Yeah, so for medicinal purposes, you would need to speak to your doctor, and doctors are able to prescribe medicinal cannabis. If your doctor wants
do you
to,
give them a reason, or
I would imagine your doctor would
can I
ask
just
for
get
a
my
region.
notepad first thing? I just need to write some.
So I'd imagine your doctor will ask for a reason. And actually, one of the big things that Helios is doing this year is we are an educational sponsor of a platform called mcinfo.com. And that is a platform for prescribers, because a lot of doctors out there are getting questions from their patients. And as part of their duty of care, they're saying, well, I need to learn more about this myself before I can prescribe it to someone else. So if you want to speak to your doctor about it and they want to learn more about it, I would recommend that mcinfo.com. And that platform actually has a part which is only available for healthcare professionals, so the public can't access it. And that's got all the information that they would need to be able to confidently prescribe medicinal cannabis for different indications.
So when you raised $15 million, what was the state of the legality of medicinal products at that time? Like, were the investors just like, we can see it coming from overseas, we're definitely going to get it here, we'll pump in this money?
Yeah, so we could see already that it was coming to New Zealand, but the initial investment rounds were undertaken before the medicinal cannabis scheme was here. So, and we actually started building out our facility before it was clear exactly what the regulations were going to look like. So that was a leap of faith that the founders and the original shareholders took.
Cool.
Talk a bit more through the progress. So we've had a couple of investment rounds. You can see there's some other phases. We finished phase 1 of our build, so of our facility at the end of last year. And what we've been up to this year, so this year is a very big year for us. The biggest milestone we've had, as we talked about, was that GMP license to manufacture medicines, which means that we can now start our commercial manufacturing processes. Before that, we've been doing a lot of work commissioning our facility, ramping up our processes, doing R&D work, but now we're able to move into that commercialization phase. And to give you a bit of an idea, this is us. This is Helios Therapeutics. So we're an 8,800 square meter site. Essentially what we've done is we've built a factory inside a factory. And so we have our grow rooms, which are all completely controlled. And on top of those sit almost a whole another factory, which is the HVAC and building management systems. We also then have our extraction units as well as our processing and lab areas inside this building that you can see here. This is what it looks like from the outside. And this gives you a bit of an idea from the scale. So on the left-hand side up there, where you can see those lines, that is the cultivation facility. And then this part here is where you've got the extraction, the processing, as well as the office and analytical areas.
So up until this point, you've obviously been growing plants.
We have, yeah.
So what have you been doing with the product generated if you can't sell it?
Yeah, so that's been R&D processes that we've been able to undertake. We haven't been able to commercialize anything yet.
So in terms of products that you're generating, obviously you want to sell them to doctors and stuff in New Zealand. How can you guarantee that they'll choose your New Zealand product over like overseas?
So we can't guarantee that. So we go out, we compete on quality. But what actually is really important for us at Helios is the journey. So our first products to market are what are known as common therapeutics. So they're a product that's taken orally, but they're also a product that anyone who is using medicinal cannabis or any doctors are familiar with. And that's really important as a first entry into market. That lets us set up our processes. It lets us set up as well our brand and our distribution channels. But actually what we at Helios are all there for is what comes next, which is that R&D pipeline where we're looking at really the different cannabinoids that we can unlock from the plant, that we can extract from the plant, as well as the different dosage forms that come with it.
Yeah, so you just mentioned there a couple of things about what the future looks like, but what else does the future hold? What else are you working towards?
Yeah, so for us, we look to produce products for the New Zealand market, but also the international markets. So we're looking at export markets and entering into those in 2022. We also have a number of research partnerships that we're working on, so collaborations with some New Zealand companies as well as some international. So you'll see some announcements about those as well, and going into the clinical trial pathways as well.
Right, cool. So got this approval. What happens now?
Yeah, so we ramp up now. So we ramp up for production. So we have the processes all there and ready to go, and we start preparing to bring products into market.
Cool. And do you need any help from us? Do you need anything?
So, I mean, I guess the help that I need from you, and I'm going to explain a bit about this. So one of our values at Helios is curious. So I would just encourage everyone out there to be curious about medicinal cannabis. If it's something that you don't know anything about, is to go out there and learn a bit more about it, ask questions and be open to learning. And I think that
develop an illness and go to your doctor.
I'm not going to ask people to develop an illness.
Pull one out. If there's one kind of starting, maybe just work with that.
And for those people who do want to learn more about medicinal cannabis or know people who are in the industry, we have an industry conference coming up in October called the MedCan Summit, and that's held in Auckland. So if you do want to get involved and know more about medicinal cannabis, that's a great opportunity to come along. All of the different industry players will be attending. So it's a great place to network and learn more about the industry and how things are progressing.
Awesome. Questions? Yep.
Do you think, like, as part of like you guys' strategy, you're doing a lot around, the IP of different, developing plant species? If there's just such a big conglomerate that can supply so cheap, but your products actually go to them by making the most unique, product that I don't know how to say it, but yeah, that focus on like, can we focus more on like the IP development around this industry, not just around selling product always?
Yeah, and I think that's where the New Zealand way of doing things is really important. It's very difficult for any industry in New Zealand to go out there and compete on a commodity basis. From a volume perspective, for us, we see that also in the medicinal cannabis industry. So it really is about providing super high quality, really safe and efficacious medicines. The best way to have those efficacious medicines is really to look at novel IP. So developing those IP pathways, looking at some of the pharmaceutical pathways that we have for dosage forms for rapid onset or for slow release, and then applying that together with a cannabinoid medicine to be able to give something that works for specific indications. So
Awesome. Next questions? Yep.
I'm quite curious. I imagine you're doing a lot of kind of breeding stuff, and there's a lot going on there. Are you interested in sort of endemic New Zealand varieties, and are you starting in that already? Or I imagine you'll get there, but what do you think about what's in New Zealand?
So that question is, are you looking at endemic New Zealand varieties?
Yeah, absolutely. So we're licensed to hold over 40 cultivars on site at Helios, and we've definitely been looking at a number of different genetics, both from international sources as well as locally here in New Zealand. Under the medicinal cannabis scheme, there is a declaration process where you can work together with people who've been growing plants and legitimize them. So something that's been grown in the black market can then be declared. And
have you got some?
That's kind of like a follow-up, which is like, how can you support the people that have been generating cannabis for the New Zealand market for the last 50 odd years or 100 odd years? And there is a community of people that have been doing that. And, it's a tricky one. But do you try to integrate that black market info? It sounds like yes.
Yeah, so absolutely we do. So my goal is, and if I look at the cannabis industry across New Zealand, is that for all of us in New Zealand, there's a real opportunity to collaborate. If we want to really take New Zealand products to the world, then we need to work together. If I look at Helios, we've got 20 people on site. If we look at the entire cannabis industry here in New Zealand, even if you add up everyone in the industry and every plant that's growing, it's still smaller than some of the big players that we're out there competing against. So where we can have a real unique advantage here in New Zealand is if we collaborate and work together.
Cool. Yep.
What implication did the referendum on recreational cannabis have for the medicinal cannabis?
So the question was, what impact did the referendum have?
Yeah, so that comes down to business models and business strategy. So for us at Helios, our business model right from the very beginning has always been focused on medicinal rather than recreational. And that's a strong view given also by all of our shareholders. So for us, it didn't change our business model or our strategy. And I can't speak on behalf of other players in the industry, but some of the other business models were banking on the referendum.
But wouldn't that recreational cannabis kind of provide free publicity for medical as well?
So wouldn't recreational provide free publicity for medical?
So I think that they're two quite separate pathways and two quite separate products. So a medicinal product, the really important thing is around accuracy, dosage rates, and consistency. So someone who is on a medicinal cannabis product needs to know that every single time that they're having that product, that it is delivering the same amount, so the same active ingredient amount, and then hence the same effect. Whereas the recreational is quite a different approach from that perspective. And so I see them as really quite two different products.
I guess it might have made people not want to try it, or there could have been something that came out of it not being approved potentially. They might not want to go down the medicinal route and know that it's a thing, or there was a question hand. Yeah.
I know you're doing research and development on what sort of ailments cannabinoids can heal, but what do you find is used for medicinally?
So what is it used for medicinally?
Yeah, so the main indications around the world for medicinal cannabis are around sleep, anxiety, and pain. There are a number of studies around the world around these indications, but there is also more and more studies coming out around some of the more minor indications. So there is clinically trialed products available for specific forms of, for example, childhood epilepsy, where we can see medicinal cannabis has clear therapeutic benefits.
Cool. Couple more. Yep.
Just I got some more of a comment about the you said go to your GP and ask about it. Well, I had that experience since 2017. told you couldn't access it, even though I was just going for CBD. And so I just encourage everybody to reach out to a cannabis clinic if your GP does turn you down, because it changed my life, and I dropped off 3 pharmaceuticals for exactly those reasons, plus one more that you didn't mention. But yeah, it makes a huge difference. I wasn't even going to be able to go and work in an office anymore. To be honest, it's made a huge difference.
So yeah, if you go to your doctor and they say no, go to a cannabis clinic.
Cannabis clinic.
Yeah.
Multiple throughout New Zealand.
Yeah, so there are a number of cannabis clinics. So I visited a number of them last week as we had our GMP certificate announced. And there's also now more and more doctors that are curious and learning more about it. And that is why we launched an educational platform. I don't know if you know, but if you Google cannabis, you'll come up with a world of different things. I'd hate to think what my Google watchdog thinks about the things that I Google, but that was a common feedback we were getting from both patients and prescribers, that they didn't have enough information to feel really comfortable and confident. So absolutely, if your GP wants to know more, then they can reach out to a cannabis clinic and they'll often be able to talk them through it. Or, like I said, we've got the info available on mcinfo.com. I can just share a little bit of a story about that. So we recently attended the GP conference in Rotorua, and we were there and talking to a number of GPs. My colleagues who were there 2 years ago said that a number of GPs would take a wide berth around the stand and avert their eyes, whereas actually now this time I would say 80% of GPs came up and say they were taking information, they were looking to actually learn and know more. So that's why I really encourage you to be curious and learn and find out more, or to encourage your GP if you do want to know more as well on that.
Cool. One more question. Yeah.
Can you talk a little about side effects? And the question or the context of that is, with traditional medicine, you have significant side effects given the synthetic kind of makeup of those. How does this sort of thing better that? And then the second part to it is, how does plant varietal development or plant selection impact the way you create ultimately the medicinal products?
So first I have to say, do you want me to share the question?
Yeah.
So the question was around side effects, and then also how different plant varietals or genetics impact our selection process. So first I have to say I'm an engineer, not a doctor. So if you want to know about side effects, you do need to talk to a professional in this area. The great thing about cannabis, though, is that we all have that endocannabinoid system in our bodies. And if you want to learn more, that's a great place to start, is learning about this endocannabinoid system. And that system, basically, in my simple engineering terms, is about restoring homeostasis in the body. So it's about restoring balance in the body. And I, in my control systems thinking, can understand that. I'm sure doctors would talk about it in a lot more detail, but this idea of restoring balance, and that's why medicinal cannabis does have a lot lower level of side effects than a number of alternatives. There are side effects, but they are very minimal. Around the different varieties, so this is when we get into talking about cannabinoids. And if we want to get really serious, we can talk about flavonoids and terpenes as well, but that's probably for another day. So the main active ingredients that you will have heard of from a medicinal cannabis plant is the CBD and the THC. In simple terms, the THC is the one that has psychoactive effects, so is often used recreationally, so to get you high, but also at the right dosage forms has therapeutic benefits as well. So both CBD and THC, either by themselves or used in combination, are used for a number of different indications. Then you get into the next level of cannabinoids. So there's plenty of those. And then, like I said, there's plenty more that are still being discovered. We also start to talk about things like terpenes or the entourage effect. And the entourage effect is this idea of when you start to put the different cannabinoids together, then actually the synergistic, the combination of them the output of that is greater than if you took each one individually. And it's still something that scientists are really investigating and trying to understand. But for example, THC, if it's taken together with CBD, the CBD actually minimizes some of those psychoactive effects. So then all of this feeds in when we're looking into genetics and plant varietal searches. When we look at our plants, we're looking at things like the different potency levels of those cannabinoids, as well as then some of the minor cannabinoids and the terpenes that we would see in those plants. So bit of a long-winded answer, but yeah, there's plenty there to learn.
Cool. That is it. Thank you, Carmen.
You're welcome.
Will you hang around if
I'll be
anyone
around
has
for a bit.
questions? Cool. Okay, so let's go straight into 1 minute pitches. I know at least 2 people that need stuff.
I'm Nick from WNT Ventures. We currently have an analyst role, if anybody's interested in being an analyst within venture capital. Our portfolio companies also have a number of roles available, from hardware development, software, so on and so forth. So if anybody's interested in a job, then please reach out.
Awesome.
Thank you. My name's Mike from a company called Carry Cone. We actually produce servers. I bought one along tonight. This is a runs full operating system, Ubuntu, Red Hat, or CentOS. If anybody knows about those sorts of things, we use them for either web hosting or for database serving or email serving. We've actually got a smaller one as well, about half the size footprint, which we call an IoT server. And on that, we use products like TensorFlow, Python, and the like for analysing images and running machine learning and artificial intelligence. So that's the second product we have. The third product we have, we've built a thing called a natural language processor for on-premise development of application software. So if you're interested in that, our business is called Carry Cone. We're based out in Henderson, and thanks for listening.
Awesome.
So hello again, everyone. Good evening, and thank you for your time. First of all, I'm Malcolm, and I've been working in an IT support company called Propellerhead in the last 2 years. So I do appreciate my current job, which I learned a lot of analytic skills with cloud computing and our customer, including Auckland Transport and the Auckland Museum and a few. So the reason why I'm here is I want to look for a job. So I love the job I'm doing, but I also want to advance my career with more interesting and challenging projects. So right now I'm doing a part-time of electrical and electronics engineering in Auckland Uni, and as a master's degree. And so anything that interests me is from electronics engineering, embedded system engineering, even the cloud application engineering. So thank you for your time, and thank you, Alice, and your team. I'm honored to be part of your team to make this happen. I just realized that COVID is still a pandemic, and I'm very lucky to be here to listen for some bright ideas from like-minded people. And thank you. Yes.
Special shout out to Malcolm. I worked with Malcolm for a couple of years. He was one of my favorite colleagues ever. So you can't go wrong with hiring Malcolm. He's amazing. I've definitely pushed him to look for another job where it utilizes his amazingness. So yeah, if you need anyone, please chat with Malcolm.
Kia ora, I'm Emily, but my pitch is on behalf of someone else. I'm mentoring a really awesome student from University of Victoria. She's studying software engineering. She's 3rd year. She interned at Google last year. And she's looking for a job at a cool hardware startup that has, awesome culture, cares about people, ideally working on a cool mission, climate change or something like that. So if that's your company, please reach out. Also, if you haven't heard, Callaghan Innovation actually give grants for interns over summer. So it also would be not cost you anything to have this awesome student. So yeah, definitely come talk to me if that is your company.
We definitely all know about those sweet grants. Thank you. Actually, I do have a pitch, and it's on behalf of ZeroJet. So ZeroJet make electric engines that they put in tenders, which are like small boats. Really cool company, amazing people involved, and they're looking for people. So you can jump on Seek, and they have a range of awesome roles. Super cool company. They want to be the Tesla of New Zealand for electric boat engines. Thank you.
Hi, I'm Danny. I've just qualified as a welder fabricator at Buckley Systems. I'm actually on the lookout for a new role. Before the apprenticeship, I worked in sales at Pulse Energy, and I actually found I really enjoyed that. So I'm looking for a role in maybe administration, something to take on a bit more responsibility, like, you know, problem solving and like trying to make things more efficient, that kind of stuff. So if anyone knows of any roles that kind of fit that description, I would love to hear from you.
Hi everyone, I'm Puri. I work for a company called Flexware. We're an engineering consultancy, and we provide hardware, software, and firmware solutions. So if you're doing a project and you need those expertise, happy to have a chat. We are also growing, so we are looking for some technical people, and then at the moment we're hiring for a project coordinator. So if you want to work for a cool employee-driven company, reach out.
Hi, slightly awkward because my current boss is here, but I'm Sarah. I've been with Stretch Sense for the past 8-9 years, so I've gone through the up and down roller coaster. I'm actually on maternity leave, so if anyone, startups or whatever, would like to have a chat and just dig my brain and make sure that I don't go completely mental with my little 3-month-old twins, please come and have a chat with me. I have a background in biomedical science, operations, manufacturing. I have hired probably 350 people in roles all over the place, and currently, or previously, most previously, been setting up companies in the UK and the US for Stretch Sense. So kind of got a bit of everything. If anyone just really needs to do a brain dump, please come and have a chat.
Awesome. No? Okay. Well, that's it. Thanks for coming, everyone. And I hope you met at least like 1 or 2 people that you've never met before. If you haven't, there's still time. And we'll have another event in a couple of months. I don't know what the theme is, but I will send out an invitation so we can potentially have a site visit at Mint, maybe Helios as well. That would be really cool. Thank you for coming, everyone.